I started out wanting to do some insect collections with my introduction to biology class so that I could then, the actual physical insects could be used at the University of Iowa for an insect class, for an entomology class. And so I was kind of excited by that because it wasn't just something my students could do that I was integrating as an outdoor activity for my non-majors biology students, but I also was partnering with the University of Iowa. And so that was kind of a nice segue into connecting the students and also visiting we went to 3 different sites. We were doing sort of a biodiversity survey of 3 different sites. One was a brand new prairie site, one was an established prairie site, and one was kind of more of a wooded prairie site that was sort of middle-aged, not yet fully established. And so we kind of had new, old, middle-aged sort of different sites. And we were comparing diversity, mostly just to talk about diversity, but then also to expose students to these places where they can actually physically go in our community. Because I often think that sometimes that students don't get outside as much as I think people should go outside. And that all went very well. We did collect a lot of insects, and I was able to provide them to the professors that needed them at the university. And so then COVID happened, and my plan for trying to go forward with that same technique, of course, completely failed because it all hinged on people being in the classroom together and people being able to travel together and meet on site together. Even though it was outdoors, in the beginning, we weren't sure how that was going to work. So reflections from the first group that got to participate were very favorable. Students, I think, always appreciate field trips and getting outside and trying to do something different. Of course, there were always the same logistical challenges of where to go, when to meet, how to get there, even though I provided maps and all of those types of things. But in general, it worked out pretty well. Then I moved after COVID, I kind of moved into kind of going back to and sort of and this is what I'm going to show you, realigning some of my my biology courses. For all of my courses, I have always done sort of a short in-class an experiment where they actually have to do science. And I have for a long time been wanting to kind of extend that and make it a little bit more. And I did some of that through COVID, not so much with my introduction to biology students, but I did it more with my Bio 2 students. So I'm going to share screen now. If I can find my right place. 1 second here. Helps if you have your screen up. Okay, so I have here just we use Talon, which is just a D2L format at Kirkwood as our LMS. And so here's my General 2 biology course from this last semester. And I have a tab for reports, and I call it step-by-step science. And if they were with me in Bio 1, then they would have also done a step-by-step science project. And so I've just kind of streamlined this and made it last a little bit longer. And I've added some things that I didn't originally have. For intro bio, I do a similar type of thing. And for bio, I do Bio 1, I do a similar type of thing. But for Bio 2, I tried to kind of extend it. And then for Bio 2, over the last couple of semesters, they've been allowed to take it home, which was a brand new thing that I had never done before, of course, because of COVID. That I had to sort of make some, I'm going to move up here, sorry, make some amendments for students who either were sick, if you're trying to do if you're trying to do a project and you're in the classroom. I was able to still meet for labs at Kirkwood. We were able to meet for labs. It was very challenging because we had a couple of different options. I'm sure, like all of you all I know have been challenged by the last couple of semesters. We could either be all online for all of our classes. I felt very strongly with science classes that seemed kind of challenging for me, and I didn't want to develop all brand new online content for all of my classes. And I teach 4 different preps, so that's just a lot of work. So I had developed some online things, of course, for past when I go to a conference or something like that. But I didn't have a full suite of an entire semester's worth of online content for all of my labs. So I was aggressive about just staying with face-to-face labs and wearing masks and doing all of the good social distancing that we had to do. What that meant then was that we either had to do half as many labs, only do labs, have a group and a B group, and do A group one week and B group the next week, and they both do the same lab, or have A group doing the lab in one room and B group doing the same lab in another room at the same time. And so that's what I opted to do. So I just ran back and forth between two rooms. And by the end of the semester, there's always a loss of students drop, or people got sick, or something like that would happen. And so it sometimes worked out that I was able to actually squeeze everyone into one room with our social distancing. So probably 3/4 of the semester was 2-room back and forth flippy floppy. So I'm just trying to describe kind of the challenge of trying to do some of the same labs. But this lab is a semester long assignment. And I actually have talked about this before, and my colleagues are probably rolling their eyes right now because they know my thing. There's an overview. We have 5 different assignments throughout the semester. The first assignment is they have to go to the actual, digital library. Sometimes they ask me physically, do I have to go to the library? And that was really good for me to quantify or qualify that there was a difference between going to the library with your body and going to the library online. I hadn't really thought about that. And so they go to the library virtually. They find a couple of articles, the organism, and of course we talk about this in class. The organism that they're going to be using for Bio 2 is Brassica rapa, the Wisconsin fast plants. They have to sort of identify what are the plants used for, what are they related to, how important they are as food crops, challenges with climate change, drought, desiccation, salinization, all of these types of things. And we have a relatively robust conversation, usually in lecture. But I had to organize a special Zoom class, Zoom meeting, to have some of that conversation this semester, since we weren't having regular lecture. And then they have to compose a short literature review. So this is sort of proving that they can find scientific information. This is proving that they can now read the scientific information and understand in general what it says. Of course, many of these articles are very challenging. From my work with Pulse, one of the things that Pulse had identified as challenges for science students was just being able to find and read scientific information. And most of my students are going into some form of health science. And so I remind them that, they have to stay up on their own current information, whatever aspect of health science they're going into. There will be new information coming out all the time, current research. So being competent to be able to understand what the research says for their specific field, but then also clients or patients are going to ask them for information. And most clients and patients come well armed with their WebMD information, and being able to share something that's really going on and share it in a sort of layperson way is important. So being able to kind of read the stuff and then understand it and digest it and reproduce it verbally for someone who doesn't understand it as well is important too. Then we do an experimental design project where they have to come up with a plan for what they're going to actually do. Again, this was always a little bit easier because it was happening all on campus. Students were coming to class regularly, and so that made that a little bit easier, managing the growth of their plants, anywhere from 2 to 6 weeks, depending on what they were doing. Then they have a draft report that they have to submit. Then they submit they share a copy of their draft report with a classmate, and they do a peer review. And this is taking place all over throughout the entire semester. And then, of course, they turn in a final draft. So the thing that I added some statistical options here that I've used in the past, but then had kind of moved away from just because there were so many other moving parts. And then the other thing I added was this, which was like, I kind of had one of those face to palm moments where I realized this whole time I've been doing this, but I've never asked them to actually like share their data journal with me. And so they actually this semester and last semester had to do a photographic journal. So taking actual photographs of their plants. No matter what I say, every semester somebody's plants die from not being properly watered. And of course, we all know, we learned in kindergarten that plants need water. So I tease them strongly about how ridiculous that sounds, but somebody's plants are going to die. And of course, somebody's plants always die. But this photographic journal helped them kind of then mentally remember to, if they did it, mentally remember to water their plants, take care of their plants, measure whatever facet of information they were collecting for their data. So there's a folder for each of these, that little introduction there. You can see there's the actual full assignment, which this is the same kind of thing I'd always had, of course, but I had most of it by paper, and we would go through it in class. As a Gen Xer, I was really fond of the paper. As an environmental science teacher, I'm trying really hard to move away from the paper. So that was kind of what happened for this last semester. Taking stuff home was, I found actually students, if they were able to and had a space to do their setups at home, was actually better to some degree, because when your project is right there in your space, you know, you can't not look at it or be curious about it. And students' reflections were really interesting. The ones that got to take it home, you know, I think we're just more curious, more interested. And even without COVID, I'm still thinking about trying to maybe do some of that. I don't know how that would work, and it's frustrating for my lab coordinator who lost a couple of lamps. A couple of lamps didn't get returned, or something got broken. But in general, I'm curious about how that might work. It's challenging from the standpoint of, you know, if someone's control group dies, then of course they can't just use somebody else's control group if we're all growing in the same place and same time. But students had such interesting experiments in the past. A couple of students have wanted to do things with music, exposing the plants to different types of music and measuring different growth measurements, height, number of leaves, number of flowers, all kinds of different things. And you know, there's a lot of good research out there. There's some good research out there about different types of music and grape growing, and then just talking to plants and how plants respond to sound. And so a student wanted to, you know, if they want to do those kinds of things, in the past, I'd allowed them to take things home, because you can't have 3 little MP3 players all in the same place. First of all, it's going to pollute the plants that are only supposed to be receiving one sound. And then from another component, it's something that could be stolen, right? If it's in a place where we're all kind of have access to. So students in the past for special things have had an opportunity to take things home, but this time it allowed students just a different opportunity. And the cool thing was then their kids or their husbands or their roommates also got invested and interested. And they reflected that, you know, my roommate helped me measure this and made sure that I took the picture from the right angle, or my kid helped me water it, or and so that part was kind of an interesting thing for me to learn, that some kind of activity that you could send home for people to do might be kind of a cool thing. So that's about as much as I have to share. I think I'm at my time anyway. I didn't think I'd be able to talk that long, but there you go.
When you mentioned the music, I was envisioning the Ghostbusters, where they're playing music for a toaster and seeing how it responds. But anyways, cool stuff. Going back to the insect collection, do you think you're going to try that again once?
Yes, definitely. I have actually reached out already to the University of Iowa professor that I was working with, Dr. Andrew Forbes, and we already are making a plan for how it's going to work now that we don't have to have COVID restrictions. So yes, I am planning to try that again this semester. My only sadness is that we've basically missed a whole time the new prairie. Um, so my curiosity sort of side project, this professor and I were actually kind of curious about what sorts of velocity we'd find even with these auction. And we're interested, probably not going to see would change in a newly established prairie. So kind of missing year one when prairies get mowed and just not having a hole in the data is frustrating, but I'm sure I'm not the only person that's been frustrated by COVID.
And then the professor that's doing this, is that going to be are they eventually going to try to publish some study out of this? And will your students get co-authorship?
I don't know about that. We have not had those kind of conversations at this point. He was just desperate. So he runs his entomology class in the spring, and I have 2 sections of intro bio usually in the fall, which meant that I had, warm bodies who could actually just go out and collect insects, which allows me then the opportunity to talk about insect collection, different methods, and I had done some, I took herpetology, but I didn't take entomology. So I need different pit traps and beetle traps, mist nets, and all these different kinds of ways to collect organisms. So that was good for me too, to sort of be reminded of how things work. And I didn't know that we have carrion beetles and dung beetles in Iowa, like legitimate dung beetles, which is kind of cool. So my learning certainly of being exposed to some of those things, and then just sharing with the students that there are all of these different methods, trying to make the insect ick factor, less overall, was kind of really just the goal for me. And then again, exposing them to the prairies and the locations where they could actually go and hike or be outside. Since it's just a non-majors bio class, it was really more discussion about biology and how important insects are, of course, for biodiversity. Whereas he was using the bodies then in his entomology class for basic identification. So yep. So we'll see. It was really just kind of our first attempt because of these workshops. That was what had caused me to reach out to him and try to develop some kind of relationship. And then COVID kind of interrupted just sort of where could we go from there, right? Our kind of the next thing that we were starting to look at was the biodiversity development of a newly established prairie compared to an existing prairie.
Sure.
And so that's where I was. Prairies and biodiversity are kind of my thing. So that's where I was excited to see sort of what would happen. And he had shared with me his list from the insects we did collect, and the diversity, was pretty much as correlated as what you would expect. The new prairie had very low diversity, and the more established prairie had more diversity, and the ancient older prairie that's like 30 years old had, of course, the most diversity. So,
right.
That correlation is not causation, I realize, but that was a good translation that I could share with the students.
For sure. Angie, it looks like you've got your hand up.
Yeah, I mean, I was, again, with the insect project. Number one, would there, what are the identification, are they doing barcoding or just at the University of Iowa?
They are doing barcoding, but they weren't doing barcoding at the point that we had the opportunity to work together. And I would love to do the barcoding. That would be really great. But we just haven't gotten that far yet.
Right. Yeah. And do you think that I'm guessing they probably have an upper limit on the number of sites and samples that they want to deal with at one time, but would the person be open to additional collaborations or not?
Oh, I think so. I'd be happy to pass his information on to you for sure. Yeah, absolutely.
Okay. Thanks.
Yeah.